Hello, everyone, and welcome to today's webinar, Unveil the Inflammatory Response with Novel Assays for Protein and Gene Expression. I am Megan Pascal of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Sigrun Badrnya, PhD, Manager, Product Development, Life Sciences of Thermo Fisher Scientific. Sigrun, you may now begin your presentation.
So thank you for the introduction and welcome again to this webinar, which will provide important insights into novel highly multiplexed protein and gene expression assays to study the inflammatory response and to support your research. As you well know, inflammation is a crucial part and host defense mechanism of the immune system, protecting from infection or injury. And the figure on the left highlights the complexity of the immune network that involves a variety of different cells and molecules, including cytokines, chemokines, and other soluble factors that are all critical players in shaping local and systemic immune responses. However, inappropriate regulation resulting in an imbalance between stimulatory and inhibitory mediators can be associated with various pathological conditions, ranging from inherited monogenic disorders, such as the primary hemophagocytic lymphohistiocytosis, as illustrated on the right, a rare genetic defect which impairs the cytotoxic function of certain immune cells, while a hyperinflammatory condition with overactive immune cells and excessive release of inflammatory soluble factors, as summarized below, might also occur as a side effect of therapies, including CAR T-cell therapy, or as a complication of infections such as induced by SARS-CoV-2. So inflammatory related disorders and their triggers can be quite heterogeneous, and consequently there is great interest in uncovering the cellular crosstalk and the complex but coordinated interplay between all these factors to better understand the mechanisms underlying various inflammatory conditions and disease states. In this context, RNA and protein biomarkers can be effective indicators of these processes, and therefore their efficient and accurate quantification by appropriate high-throughput tools very useful to explore and study all facets of the immune system. InvitrogenTM Plex and ProcartaPlex assays make use of the established multiplex technology on the Luminex instrument platform, enabling a unique multi-omics approach by providing a high-throughput solution for the measurement of both RNA and protein expression levels on one instrument to support immune monitoring, cytokine profiling, and to accelerate the discovery and verification of biomarkers and targets across key research areas. So notably, both applications allow for accurate and simultaneous measurement of up to 80 targets in each well of a 96 or 384 well plate from a variety of sample types from different species, and with just simple requirements for sample preparation. So these assays, which are on the one hand hybridization-based QuantiGene Plex assays for RNA quantitation from a single lysate or even from complex tissue samples, and on the other hand, ProcartaPlex immunoassays for protein quantitation from serum, plasma, cell culture supernatants, and many other biofluids, are reliable, scalable, and highly customizable tools for high-throughput protein and gene expression studies. So as mentioned, both assays make use of the XMAP multi-analyte profiling technology, which basically relies on proprietary magnetic microspheres that are color-coded with combinations of red and infrared fluorophores, resulting in unique and spectrally distinct bead sets or bead regions as illustrated by the schematic, and that can be classified by any Luminex instrument. And as outlined here, ProcartaPlex assays enable protein target detection via highly specific antibody pair binding, similar to a traditional ELISA, by utilizing these color-coded and antibody-conjugated magnetic capture beads to specifically bind an antigen, as well as a biotin-conjugated antibody-mediated detector using a streptavidin phycoerythrin as a fluorescent reporter molecule, which is compatible with all Luminex instruments. With regards to signal detection, the optics in the compact analyzers excite the internal bead dyes to identify each of these microspheres, as well as any reporter or detection dye captured, which allows for the quantification of individual analytes bound on each microsphere. The workflow, as shown on the right, further illustrates step by step how it works. So each bead set is assigned to a different target analyte and coded with a target-specific antibody. And these differentially dyed capture bead sets are mixed and added to a sample for a 2-hour incubation, allowing the analyte a specific capture of multiple targets from a single sample in parallel. And following a wash step, the captured protein biomarkers are incubated with a mix of biotin-elated and analyte-specific detection antibodies to form an immunocomplex. And after another wash and incubation with streptavidin phycoerythrin and removal of excess fluorescent reporter molecule, the sample is finally ready for readout and analysis on one of the Luminex instruments. Taking a look at the QuantiGene Plex assays that combine both the XMAP bead-based multi-analyte profiling technology and branch DNA technology for target hybridization and direct measurement of RNA transcripts via signal amplification rather than target amplification. And as illustrated here, the ELISA-like workflow is a straightforward 4-step process, starting with the preparation and lysis of samples to release and stabilize RNA from cultured cells, whole blood, virus, bacteria, to more complex tissue samples from plant or animal origin, followed by a target hybridization step upon overnight incubation with target-specific probe sets on Luminex capture beads that are comprised of specific capture extenders, label extenders, and plugging probes. Finally, in the third step, sequential hybridization of pre-amplifier, amplifier, and biotin-elated label probes build a signal amplification tree, and using a streptavidin phycoerythrin as a fluorescent reporter molecule, the signal can be finally detected on one of the Luminex instruments that is used for readout and analysis. The value of pairing these two applications provided on one instrument is further highlighted by a study previously published in Methods. So complementing ProcartaPlex immunoassays with QuantiGene assays to measure both proteins and RNA levels from the same sample provides a more comprehensive data set which can help to find the interrelation, particularly in complex biological processes or disease models. So to examine if protein levels reflect the corresponding gene expression levels within a single sample run on a Luminex system, isolated cells have been stimulated with LPS for up to 72 hours, and the supernatants profiled for protein levels on a ProcartaPlex 65-plex panel, and a cell lysate used for measuring RNA levels on a matching QuantiGene Plex panel on a Luminex system. On the right, the results illustrated for total protein quantitation indicated by the bar graphs, and the normalized gene expression analysis indicated by the line graphs for 6 different target analytes that demonstrate similar trends with corresponding gene expression levels and protein translation for IL-1 beta, TNF alpha, IL-6 and MIP-1 beta in figure A, while figure B below shows also some examples of inconsistent trends with non-matching changes in RNA and protein levels for IL-16 and trail, pointing out that mRNA expression may not always translate to the protein level, and so the measurement of our mRNA or protein expression levels alone may not tell the complete story. Overall, this example demonstrates that a single sample can indeed provide both gene expression and protein profiles, which enables more comprehensive exploratory experiments and providing a more holistic view on a particular research topic. Another published example demonstrating the combined use of QuantiGene Plex and ProcartaPlex assays is a research study on siRNA-based identification of targets associated to inflammatory bowel disease in a monocyte-derived dendritic cell models. Here, QuantiGene Plex and ProcartaPlex multiplex assays have been used to measure gene and protein expression levels of anti-inflammatory IL-10 and pro-inflammatory P40 levels amongst other targets. Primary testing using a specific IBD library identified siRNA pools that preferably reduce the P40 secretion and increase the IL-10 secretion and or IL-10 P40 ratio. And in-depth analysis finally led to the selection of 10 IBD-related targets that are further verified as potential anti-inflammatory drugs in this case. So taking a closer look at the QuantiGene Plex assays that are also available in the 384-well format, enabling even higher throughput multiplex gene expression analysis and allowing for more efficient compound testing to accelerate biomarker and drug discovery studies. Here is a detailed overview of the complete workflow for 384-well assays, including information on the hands-on and incubation times for day 1 and day 2 of the protocol. So while the hands-on time of 1 hour on day 1 and of less than 1 hour on day 2 is fairly minimal, there are still various steps in the workflow, particularly on day 2, that require the transfer of samples or washes between 3 times 1 hour incubation, which can be considerably simplified by integration of automated equipment. So automation and batch processing options are readily available for this assay format to further decrease hands-on time while increasing the sample throughput. So with higher throughput and an increasing number of data points, software solutions for streamlined analysis are getting more and more important. And there are 2 software solutions available to support QuantiGene Plex data analysis. On the one hand, the QuantiGene Plex data analysis app, which is free software available through Connect, the Thermo scientific cloud-based platform, with an easy-to-follow workflow that provides also customization capabilities for normalization and analysis of gene expression data. The data collected from QuantiGene Plex assays can be furthermore also exported into the transcriptome analysis console software for even more advanced data processing and graphical analysis, including heat maps, volcano plots, or pathway analysis. On the following slides, I'd like to share a few published examples of how QuantiGene Plex assays have been used for high-throughput analysis in different research fields. So in this first example, a QuantiGene high-throughput assay has been used for biomarker studies in iPSC-derived retinal pigment cells. In this case, the research group started out by establishing and thoroughly validating a QuantiGene Plex assay in the 96-well format before scaling up to run it in a 384-well format to increase the throughput and also decrease required sample input. They found a significant correlation of multiplex gene expression data from QuantiGene Plex assay with standard qPCR measurements as shown here in the correlation plots for the 384-well format, and concluded that this high-throughput assay provides the basis for the compound testing studies and for further exploring future treatments for several retinal degenerative diseases. The value of multiplex analysis using high-throughput QuantiGene Plex assays has been further confirmed by another research study as shown here on cytokine-driven goblet cell metaplasia in the lung, which is a hallmark of chronic lung disease and lacks treatment options. So using a high-throughput QuantiGene Plex 384-well assay for a compound library analysis in 3D bronchospheres, the research group was able to test almost 5,000 compounds and identified that Notch2 is required for goblet cell metaplasia in the lung. More specifically, they concluded that inhibition of Notch2 prevents goblet cell metaplasia induced by a broad range of stimuli, and are now following up to explore potential future therapeutic avenues for this condition in airway diseases. In a third example, the QuantiGene Plex 384-well high-throughput assay has been used in a research study to measure specific gene expression-based signatures in order to identify functionally similar proteins. In this context, assessment of the technical reproducibility using a housekeeping target furthermore revealed a high level of correlation between replicates, concluding that the QuantiGene Plex platform has distinct advantages and is a valid method for high-throughput gene expression analysis. So switching or turning now to ProcartaPlex assays for highly multiplex protein expression studies. The portfolio of ProcartaPlex panels has just been further expanded, now offering the new immune response panel, which is currently the largest multiplex protein assay available on the Luminex XMAP-based system that enables the simultaneous measurement of 80 key soluble factors in a single well to study the immune function and providing a more complete picture of the immune response network. Performance evaluation of this immune response ProcartaPlex panel in untreated human serum samples and pre-treated plasma samples revealed the differential expression pattern of these 80 targets as illustrated in the bar chart. And moreover, side-by-side comparison of sample concentrations detected in human serum and stimulated plasma samples between a small plex, in this case a 9-plex chemokine ProcartaPlex panel, and the human immune response 80-plex panel, as shown here for selected representative analytes, human MCP4 and CCL1, demonstrate consistency in both large and small scale multiplex panels, enabling high content analysis without compromising the assay performance. Further regression analyses that have been performed comparing the small 9-plex and the 80-plex panels reveals a high correlation between large and small scale multiplex assays with an R-square value greater than 0.9 for both selected targets as illustrated here, which makes it easy to switch between small-plex and high-plex panels while ensuring reliable and reproducible results. Besides performance comparison between the human 65-plex and the 80-plex panel in human plasma samples, furthermore demonstrate also highly correlating sample values, as shown here for human MDC as representative target that underscores the consistency also across high-plex panels. So how are high-throughput multiplex protein assays used to support research and preclinical studies? On the following slides, I'd like to also share a few published examples of how ProcartaPlex multiplex panels have been used for immune monitoring across different research fields, including SARS-CoV-2 research, as shown here in a first example on the left, in a publication in Nature, where the Th1, Th2 cytokine 11-plex mouse ProcartaPlex panel has been used in the preclinical development of 2 vaccine candidates by BioNTech Pfizer to analyze the T-cell mediated antigen responses in mice following vaccination. As well as in a second example, as shown on the right, where the human immune monitoring 65-plex ProcartaPlex panel has been used in a cross-sectional exploratory study to determine specific cytokine profiles in peripheral human blood during the early onset of COVID-19 infection to ultimately help identify and develop innovative treatment strategies against SARS-CoV-2 infection. ProcartaPlex panels have also, high-plex panels in particular, have also been widely used for immune monitoring in the cancer research space, such as shown here, where in total 59 chemokines, cytokines, growth factors, and soluble immune oncology checkpoint molecules have been measured in plasma samples collected from individuals with metastatic diseases to determine the response to immunotherapy. Following a discovery and verification phase, finally a 2-cytokine signature composed of RANTES and PD-L1, as shown in the scatter plots below, was found to be associated with increased disease control rate and survival, which underscores the predictive value of these targets for the stratification of individuals who may benefit from a certain treatment. Moreover, as shown on the right, the human immune monitoring 65-plex ProcartaPlex panel has also been used to identify serum cytokine signatures to help stratify individuals with systemic sclerosis and to differentiate between high and low risk groups, as well as to identify also distinct cytokine expression profiles from human serum and exosome samples, as summarized on the left, dependent on different isolation methods, and to further evaluate exosomes as potential blood-based biomarkers for metastatic processes and more advanced tumor stages. So altogether, these examples underline the value of ProcartaPlex multiplex assays as efficient, high-throughput tools for immune response profiling in a wide variety of sample types from different species and across different research fields. To accelerate protein profiling and research even further, ProcartaPlex 384-well assays enable multiplex protein analysis also in a high-throughput format with optimized reagent formulation that has been specifically developed for smaller reaction volumes in the 384-well flat-bottom plates that require less sample input compared to a 96-well format, however, without compromising the sensitivity and performance of the assays. As shown here, ProcartaPlex 384-well assays are available in 2 different formats. One, on the one hand, as a liquid formulation of premixed bead sets that are provided in a single vial for an extensive selection of analytes from a variety of different species, as well as an exclusive ready-to-use format with plates and pre-dispensed lyophilized beads for workflow convenience. Taking a closer look at the workflows for ProcartaPlex 384 well assays with liquid versus lyophilized beads. So as compared to the liquid beads option that provides premixed bead sets that are to be pipetted on 384 well flat bottom plates, the 384 well assays with lyophilized beads come with pre-dispensed capture beads lyophilized on 384 well flat bottom plates, which eliminates the bead dispensation step in the general workflow as outlined here, thereby minimizing bead dispensation errors, and also reduces the hands-on time, which makes the workflow even more convenient. Comparison between the 96 well and 384 well liquid and lyophilized bead formats, as shown here, revealed a similar performance of standard curves from the same standard mix lot in different product formats, as illustrated here for 3 selected analytes, ensuring consistent results regardless of the ProcartaPlex assay format. Furthermore, when comparing sample values between the 96 well and 384 well liquid and lyophilized formats for selected analytes, furthermore, high correlation of sample concentrations across the ProcartaPlex formats demonstrate reliable and reproducible results. This is further confirmed by regression analysis obtained from human plasma samples between the 96 well and the 384 well liquid format, which furthermore shows excellent sample correlations with an R-square greater than 0.9 for these representative analytes, making it easy to switch between 96 well and 384 well formats, ensuring consistent results. And finally, high sample correlation could also be observed between the two ProcartaPlex 384 well formats that clearly demonstrate equal performance of assays with lyophilized and liquid bead formulation, as illustrated here for the 3 selected analytes on this slide. Importantly, to enhance also data analysis, a free cloud-based software solution is available on the Thermo Fisher Scientific Connect platform. So with the InvitroGen ProcartaPlex Analysis App, which enables analysis of ProcartaPlex multiplex data required on Luminex and BioPlex instruments from both 96 well and 384 well formats, featuring 4PL and 5PL curve fit optimization, group-wise statistical and heat map analysis, as well as the export of detailed reports, including also publication quality images and data. So to conclude, the InvitroGen ProcartaPlex and QuantiGene Plex multiplex assays offer a unique high-throughput solution with the highest flexibility to measure both gene and protein expression levels without changing the instrument platform to uncover the immune response network in all aspects. And besides pre-configured, ready-to-use panels, the new custom panel configurator tools available on the website for ProcartaPlex and QuantiGene Plex assays help to design your own panels up to a maximum size of an 80-plex and to mix and match combinations of available targets appropriate to your research needs. Notably, the 384 well format combined with the capabilities of the Luminex IntelliFlex or FlexMap 3D systems enable even higher throughput applications and allow to simultaneously interrogate large sets of RNA or protein targets with smaller sample input, however, while maintaining consistent performance as compared to the 96 well format, and as shown by comparison data. To further shorten your path from sample to discovery and accelerate your research, you can take advantage also of custom services that are provided for all the assay platforms by outsourcing your assay development, validation, or sample testing. So with that, I would like to thank you all for your time and interest in this presentation, and I'm ready to take any questions now.
Thank you, Sigrun, for your informative presentation. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box and click Send. We'll answer as many questions as we have time for today. Let's get started. Our first question is, can I trust the results obtained from a huge multiplex panel like the 80-plex?
So thank you for the question. Yes, indeed. So the scalability with reproducible performance has been extensively evaluated and demonstrated by comparison data of a small plex panel versus the 80-plex ProcartaPlex panel, as well as between also high plex panels, as shown for selected targets of the 65-plex human immune monitoring panel compared to the 80-plex immune response ProcartaPlex panel in the presentation. So really confirming that you get consistent results with any plex size.
Great, thank you. Our next question is, when would you recommend the use of multiplex assays and when would you use ELISA?
So as mentioned, multiplex assays are really ideal research tools that can efficiently support biomarker or drug discovery studies. So for analysis of larger compound libraries, for cytokine profiling, immune monitoring, and many other applications. So basically, whenever high throughput or more comprehensive analysis of a broader set of targets is needed, while the ELISA is still the gold standard of immunoassays and are powerful tools for single analyte detection and for verification. And notably, a particular benefit when choosing the different formats and platforms, such as high plex, small plex, or single analyte assays from the same provider, so from Thermo Fisher Scientific, is whenever technically feasible, the same antibody pairs are used for development of assays for one analyte, which makes switching between the formats and the platforms easy while maintaining also consistency of results. So allowing to really choose the appropriate tool for your research needs and for answering a particular research question.
Thank you. Our next question here is, what do I need to run a ProcartaPlex or QuantiGene assay?
So you get everything you need to run a ProcartaPlex assay or QuantiGene Plex assay directly from Thermo Fisher Scientific. And as a Luminex partner, this also covers the entire range or portfolio of instruments for multiplexing readouts, including also the IntelliFlex, which is the most recent and advanced addition to the XMAP line of Luminex instruments. That is also now available in a configuration with a dual report detection system and capabilities allowing for the simultaneous measurement of two parameters per bead region, so from the same bead via an additional violet reporter laser and detection system.
Great, and it looks like we have one more question here. Are 384 WOW assays with localized beads available for all PPX panels?
So currently available catalog products or panels, ProcartaPlex panels in the 384 well format with the lyophilized bead option include the human immune monitoring 65-plex ProcartaPlex panel, the cytokine, chemokine, and growth factor 45 human ProcartaPlex panel, as well as the immune monitoring 48-plex mouse ProcartaPlex panel. However, you can build your own ProcartaPlex 384 well panel, as mentioned, in a mix and match style, using also the panel configurator tool and select the lyophilized bead or lyophilized on plate option for the respective bead formulation.
Great. It looks like that brings us to the end of our Q&A session today. Thank you so much, Sigrun. Do you have any final comments for our audience today?
Thank you so much for your interest in this presentation, and hope to see you the next time. Thank you.
Yes, thank you again, Sigrun, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank our audience for joining us today and their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with colleagues that may have missed today's live event. Until next time, goodbye.
Bye.